Hello, everyone. Welcome to Future Doc Explains, where we are responsible for making science easy. So, in today's video, we're actually going to talk about shuttles, and we're going to talk about two types of shuttles specifically. We're going to talk about the Glycerol Phosphate Shuttle, and we're also going to talk about the Malate Aspartate Shuttle. So, what are shuttles? So, I'll just scroll down so you guys can see the entire photo for today. So shuttles are responsible for bringing reducing equivalents from the cytosol to the electron transport chain. So in different videos or just from your learning in school, you probably have learned about the process of glycolysis. And you probably have also learned that the entire purpose of glycolysis is to make ATP and also to make reducing equivalents in the form of NADH. And eventually that NADH will need to go to the mitochondria so it can participate in the electron transport chain, where NADH can eventually be a part of the electron transport chain to make ATP. So this is similar to what happens in pyruvate dehydrogenase and the citric acid cycle. The only difference is in pyruvate dehydrogenase and in the citric acid cycle, those reactions are already occurring in the mitochondria, so the NADH can just easily go on and be added to the electron transport chain. In glycolysis, though, NADH is actually being produced in the cytosol, since that is where glycolysis occurs, and it needs some type of way to get it to the mitochondria. So we have two different shuttles, this Glycerol Phosphate Shuttle, and we also have this Malate Aspartate Shuttle. And this will allow for these reducing equivalents, so specifically this NADH, to go from the cytosol to the electron transport chain. So this is really used for glycolysis. This is really used for NADH produced by glycolysis in order for it to get back to the mitochondria for the electron transport chain, but it can be used in other instances. So NADH cannot go through the mitochondria. So since NADH is not permeable to go through the mitochondria, that is the whole purpose of us having these shuttles, to bring these reducing equivalents more easily into the mitochondria for the electron transport chain. So the first shuttle we're going to talk about is the Glycerol Phosphate Shuttle. The way the Glycerol Phosphate Shuttle works is we're going to have two reactions happening at the same time in the cytosol. So it makes sense for us to be starting in the cytosol, since we are trying to take NADH that's in the cytosol into this inner mitochondrial membrane. So the first thing that's going to happen in the cytosol is we're going to have NADH being converted to NAD, and at the same time DHAP is being converted to G3P. So you guys probably have heard of DHAP and G3P in terms of glycolysis. So these are those same molecules. So glycerol 3-phosphate dehydrogenase, this enzyme in the cytosol, is going to be responsible for taking NADH to NAD, and also for taking DHAP to G3P. So from here, what will happen is that NAD will go on to be reused during the glycolysis pathway or just through various other pathways. In the meantime, that glycerol 3-phosphate is going to come inside of the cell. Not inside of cell. That glycerol 3-phosphate is going to come inside of the mitochondria. And in the mitochondria, that glycerol 3-phosphate is going to go from FAD to FADH2, which then will allow for that G3P to be converted back to DHAP. The enzyme that does this is glycerol 3-phosphate dehydrogenase, specifically mitochondria. So that is how the Glycerol Phosphate Shuttle works. It's taking NADH and essentially remaking it in the form of FADH2 inside of the inner mitochondrial membrane. And so from here, this FADH2 can go on to be a part of the electron transport chain. So NADH goes to NAD via glycerol 3-phosphate dehydrogenase in the cytosol, at the same time as DHAP goes to G3P. G3P can cross that mitochondrial membrane, so it will come inside of the mitochondria, and G3P will get converted back to DHAP, and at the same time we have FAD going to FADH2 via the glycerol 3-phosphate dehydrogenase enzyme in the mitochondria. So NADH reducing equivalents from glycolysis are converted to FADH2 inside of the mitochondrial membrane. So the next thing we're going to go through is this Malate Aspartate Shuttle. So the malate aspartate shuttle is a little bit more complex, but this is actually the shuttle that is preferred to be used because unlike glycerol 3-phosphate, instead of having NADH be converted to FADH2, NADH is going to be converted right back into NADH. So it's like you get what you started with, whereas in the glycerol phosphate shuttle, you don't get what you started with. So malate aspartate shuttle. What's happening here is NADH is going to go to NAD at the same time as oxaloacetate going to malate. In this process, there's actually going to be two different enzymes doing this. So malate dehydrogenase is what's taking NADH to NAD. And oxaloacetate is, an aspartate aminotransferase is what's taking oxaloacetate to malate. So NADH is going to NAD and oxaloacetate is going to malate via these two different enzymes, malate dehydrogenase and aspartate aminotransferase. And this is occurring in the cytosol. From here, malate is then going to go into the mitochondria matrix, or more specifically, the inner mitochondrial membrane and whatnot. And this same reaction is going to happen. So in G3P up here, we kind of just had it be reconverted back. Whereas over here, it's literally the same thing. So now malate is going to go back to oxaloacetate and NAD is going to go back to NADH. And they're going to use the same enzymes that they used on this side. So on this side, we have NADH going to NAD via malate dehydrogenase. And over here, we have the reverse. We have NAD going to NADH via malate dehydrogenase. And over here, we have oxaloacetate going to malate via aspartate aminotransferase. And over here, we have malate going to oxaloacetate via aspartate aminotransferase. So as you can see, the NADH that we started with in the cytosol is eventually getting remade in the mitochondrial matrix. So it's giving us those same reducing equivalents instead of converting it to some other reducing equivalents. So there is a last complicated part. So oxaloacetate cannot cross the mitochondrial membrane, although malate can. So what actually has to happen is oxaloacetate is going to get converted to alpha ketoglutarate, and alpha ketoglutarate can then cross the membrane. And then alpha ketoglutarate is going to get converted back to oxaloacetate. So I'll say it again. Oxaloacetate cannot cross the mitochondrial membrane. So in the process, oxaloacetate has to be converted to alpha ketoglutarate, and alpha ketoglutarate can then cross that mitochondrial membrane, and alpha ketoglutarate will turn back into oxaloacetate. So in order for oxaloacetate to get converted to alpha ketoglutarate, we have to put in glutamate and get out aspartate. And in order for alpha ketoglutarate to get converted back to oxaloacetate, we have to do the reverse. So we have to put in aspartate and get out glutamate. So oxaloacetate is going to alpha ketoglutarate to cross the mitochondrial membrane by the process of glutamate going to aspartate. And then that aspartate will also cross the mitochondrial membrane. That way it can do the same thing on this side. It can take that aspartate back to glutamate. That way alpha ketoglutarate can go to oxaloacetate. So in summary, for the malate aspartate shuttle, NADH is going to NAD via malate dehydrogenase, and oxaloacetate is going to malate via aspartate aminotransferase in the cytosol. This malate can then cross the mitochondrial membrane. It will go inside and do the reverse. So malate will go to oxaloacetate via aspartate aminotransferase, and NAD will go to NADH via malate dehydrogenase. Oxaloacetate cannot cross this mitochondrial membrane, so oxaloacetate has to be converted to alpha ketoglutarate, and at the same time, glutamate is getting converted to aspartate. This alpha ketoglutarate and aspartate will now both cross the mitochondrial membrane, and the reverse will happen. Alpha ketoglutarate will go back to oxaloacetate, and aspartate will go back to glutamate. And now glutamate can come back over here, and the whole entire cycle can repeat itself. I want to say thank you for coming to another video with Future Doc Explains. If you have any questions, please comment them underneath this YouTube video or reach out via email at futuredocexplains@gmail.com. Thank you.